Hello friends, Ace Botany, next episode. In this episode, we'll be discussing Cell, the Unit of Life, chapter number 8 and third part. And what all things we'll be discussing in this episode. For that, I will share the screen so you'll be getting the notes as well as content. So I will share it. I think screen came, so we can proceed. Cell, the Unit of Life, 3, chapter number 8. And what all content or what all things we'll be discussing in the episode. Initial and introduction will be there, then eukaryotic cell membrane, cell wall, endomembrane system, and introduction will be there. After that, 5 theory question, 5 NEET model question as usual with the key. Meanwhile, shorts every day I am putting at least 2 questions, preferably after 9 o'clock p.m. So when we enter into the introduction, see, comparing with the prokaryotic cell, eukaryotic cells are more advanced. And eukaryotes include all the protists, plants, animals, and fungi that we already learned in bioclassification. Now, general features of eukaryotes. So when we come to general features of eukaryotes, we can find, it's a theory question, extensive eukaryote features we are discussing, extensive compartmentalization of cytoplasm, presence of membrane-bound cell organelle, organized nucleus, presence of nuclear envelope, have complex locomotor and cytoskeletal structures, their genetic material organized into chromosomes. And these features we cannot find in prokaryotes. I think idea clear, first page is clear. Now we can proceed to second page. So when we proceed to second page, we can find all eukaryotic cells are not identical. All eukaryotic cells are not identical. It's an important question. Plant cells and animal cells are different. Prokaryote we discuss in opposite manner. Here, all eukaryotic cells are not identical. Plant cells and animal cells are different. And what are the main differences between this plant cell and animal cell? Plant cell have cell walls, plastids, large central vacuole. Animal cell having centrioles. Now, 2 diagrams, photographs you can find from NCERT figure number 8.3, plant cell, animal cell, theory question, entrance question, labeling question, different manner can be. I think idea clear. Now we can proceed to our next item, that is plasma membrane, otherwise called cell membrane. So when we say cell membrane, it is a covering for all living cell. It is so important in which the cell and outside is connected through the cell membrane. Detailed study of cell membrane was able to do only in 1950s. Reason is discovery of electron microscope during that time. Meanwhile, chemical studies performed, scientists were able to suggest structure of by analyzing human RBC. Now, studies show that cell membrane mainly consists of lipids and proteins. And the major lipid, as a question, it is phospholipids that are arranged in a bilayer manner. Lipids are arranged within the membrane with the polar head, hydrophilic, water-loving, towards outer side, and the hydrophobic, water-hating, non-polar tail towards the inner areas. The polar heads can interact with water, that is hydrophilic, which gives protection for non-polar hydrophobic tail from aqueous environment. Figure 8.4 NCERT. In addition to phospholipids, membrane also contain cholesterol. Page number 93 NCERT. Page number 93 NCERT. Now, next is an important term. It is called amphipathic nature. Amphi means both. Amphibolism, amphimix is all we studied earlier. So, amphipathic nature means these substances have both hydrophilic and hydrophobic region connected to both polar and non-polar environments. And this type of molecules play a crucial role in biological system. Example is cell membrane, amphipathic, hydrophobic, hydrophilic. And these are some diagrams. First one, NCERT only 8.4. It's a labeling question. Other is just to show how it will be. Then this one, that is based on unit membrane concept by J.D. Robertson. I think now this, just for a namesake, I drawn this diagram, but here a statement I given. Thickness of cell membrane is around 75 angstrom. How that 75 comes according to Robertson, this is explainable to it. 35. Total 75 angstrom. I think idea clear. Now we can proceed to next page. Again, a set of diagram where diagrams basically belongs to old NCERT. Just for a namesake, I shown it. First one, that is this one, David Robertson, unit membrane, 1959. Now this one, here, trilaminar concept, sandwich model. It is concerned with a scientist, two scientists, Davis and Daniel in 1935. Presently, NCERT not directly dealing with this two diagram, just mentioned it, that's all. And this is the most famous fluid mosaic model of plasma membrane, proposed by Singer and Nicholson in 1972. It's a labeling question also, famous question. So the 3-4 diagrams, just for a reference I given there, some are from all NCERT. Now we can proceed to next page. See, different models were proposed for cell membrane. Basically, all suggest that presence of mainly proteins and lipids. So mainly proteins and lipids. Later, biochemical analysis clearly revealed that the cell membrane also possesses carbohydrate. Ratio of protein and lipid varies considerably in different cell types. Normally, it is like protein iceberg in a sea of lipid. Protein iceberg in a sea of lipid. Connect with the Singer-Nicholson model. Protein iceberg in a sea of lipid. So iceberg representing protein, that is small amount. Lipid means sea is a huge amount. But in RBC, human erythrocyte, as approximately, there is a reversal of it quantity-wise. It's an important question. 52% protein and 40% lipids. Now, classification, how this has been classified. See, depending on ease of extraction, there's an important term for exam. Ease of extraction, membrane proteins can be classified as seen on the outer side, seen inside. That means peripheral and integral proteins. Peripheral proteins lie on the surface of membrane. From the term itself, we can guess. While the integral proteins are partially or totally buried in the membrane, integral, inside. NCERT page number 93. Again, the diagram I shown here. I think idea clear. Now we can proceed to next page. Theoretical chapter important for exam, theory entrance. In 1972 year is important, famous two scientists S.J. Singer and G.L. Nicholson. Model is fluid mosaic. According to this model, quasi-fluid. There you can find an asterisk mark. Quasi means semi-liquid or semi-fluid, that way. Quasi-fluid nature of lipids enables lateral means sidewise, lateral movements of proteins within overall bilayer. And this ability to move within the membrane is measured as its fluidity. Theory question, entrance question. So, lateral movements of proteins within the overall bilayer, this ability to move within the membrane is measured as its fluidity. Quasi means semi. NCERT page number 94, it is specifically given. Now, peripheral proteins are seen on the periphery, that is how the term came, on the surface of membrane. At the same time, integral proteins are partially or totally buried. So it has got some names like tunnel protein or integral proteins, which run throughout the lipid bilayer, which is also called transmembrane proteins, and their removal is difficult and requires crude methods. Again, a previous question, like treating with the detergents. So using treating or with the detergents, we can remove it by using crude methods. NCERT famous entrance point of view question, that is treating with the detergents. Now, lateral movements of protein, photograph I given there. So lateral movements of protein, lateral means sidewise, this way. Lateral movements of proteins within the overall bilayer. That photo you can observe there, same line, they move. While this is flip-flop. Flip-flop means this way. Flip-flop movement between one layer to the other layer. That is how this lateral and flip-flop differs. And this area says that some kind of research is still going on. It's a very complicated area. Proteins rarely flip-flop across a cell membrane. Lipids can flip-flop better than proteins. So lateral movement here it is flip-flop. I think idea clear. Next is importance of fluid nature of the membrane. What is its importance? It is concerned with or connected with the functions like cell growth. So fluid nature of membrane, what is importance? Connected with the functions like cell growth, endocytosis, formation of intercellular junctions, secretion, cell division, etc. Another major function of cell membrane is passage of material. In most of the case, we can find no energy is involved. And here the diagram, it is showing that endocytosis. If it is liquid, pino. If it is solid, phago. Phagocytosis, pinocytosis. Totally, it is called endocytosis. Again, previous NCERT diagram. Now we can proceed to passive transport. Passive means inactive, so no need of energy. Molecules move across the membrane, especially neutral solutes. And the procedure is simple diffusion along the concentration gradient. From where to where? From higher concentration to lower concentration. Water moves from higher to lower concentration of water. See, concentration what here means purity. From more pure water to less pure water. Movement of water by diffusion is called osmosis. NCERT page number 94. I think idea clear. We can proceed to next page. Next comes another type of transport, means facilitated transport. Facilitated means as the polar molecules cannot pass through the non-polar lipid bilayer, they need a carrier protein of the membrane to facilitate, to help their transport. Previously, a chapter was there, transport in plants. Present NCERT was eliminated, where this all explained, coming under physiology first chapter in plant side. Now comes next transport, active transport, which is otherwise called uphill transport. Some ions or molecules are transported across membrane against their concentration gradient. So that means from lower to higher concentration, normally from higher to lower concentration of water, here lower to higher concentration. This method need energy, that is ATP will be utilized. And typical example in the system is sodium potassium pump in animal cells. So you maybe studied it is an important area, sodium potassium pump, and how it is or how it become ATP consuming. See, one ATP molecule is used to move 3 sodium ions out of the cell and 2 potassium ions into the cell, crucial for various cellular activities, nerve impulse transmission, etc. So one ATP, that is 3 sodium ions out of the cell. Out of cell means normally extracellular, and 2 potassium ions into the cell, so intracellular. And that is required for various other activities. I think idea clear. Now we can proceed to next heading for us, it is cell wall. Cell wall is a structure seen outside the cell membrane. It is a non-living layer, rigid structure. It is seen in various organisms like plants, fungus, bacteria, algae, etc. I think idea clear. Now next page. So when we come to plant side, cell wall is mainly cellulose, have extra chemicals like hemicellulose proteins and pectins. Algae, mainly cellulose, have galactans, mannans, calcium carbonate as minerals. Fungus, mainly chitin, a polymer of N-acetylglucosamine NAG unit. Then bacteria, we already studied peptidoglycan cell wall. NAM, NAG residues are connected by beta-1,4 glycosidic bond. Last episode, we maybe studied it. In a plant cell, from outside to inside, what will be the arrangement of layers? Outside will be middle-lamellae, that is cementing 2 cells, plant cell. Inside or next layer to the inner side, it is primary wall. Next layer to the inner side, it is secondary wall if present. And innermost layer will be cell membrane, that is closest to cytoplasm from outer to inner. I think arrangement is clear. And these all diagrams, old NCERT just showing some representation. Present NCERT, this will not be there. Okay, I think idea clear. We can proceed to next page. Now, each layer we can explain starting with the primary wall. So, primary wall is in a young plant cell. That is why it becomes primary. Usually a single layer, and features include it is a thin elastic able to expand in a growing cell. It is able to grow by addition of more wall materials within the existing one. That is primary wall. And typical example for primary wall, important question, parenchyma cells and meristematic cells have only primary cell wall. Now comes secondary wall. Secondary wall means later. As cell matures internal to the primary wall, more layers of wall materials will be added. As primary wall will gradually diminish. Now these layers are called secondary cell wall. Along with it, thickening also takes place. Chemicals like lignin, suberin, etc. Harder, woody parts, it will be more strong. Now, after secondary wall, next layer is middle lamellae. Middle lamellae is an important item for exam. Adjacent plant cells are cemented by thin, sticky, amorphous material. It is mainly made by calcium and magnesium pectate. Here you can just see middle lamellae. That is why I put these diagrams. Okay. Calcium and magnesium pectate. When raw fruit ripens, middle lamellae become jelly-like. As a result, fruit becomes soft. Since middle lamellae act like a barrier, the cell wall and the middle lamellae may be traversed by a structure called plasmodesmata. In this figure, you can find again old NCERT, present NCERT not there, called plasmodesmata, through which cytoplasm of neighboring cells are connected. It's a beautiful diagram, old NCERT. Plasmodesmata shown there. Now comes functions of cell wall. Connected with the shape of the cell, protection from mechanical injuries. It tries to control certain pathogens. It is connected with the passage of materials. Now again connected with the cell wall, we can find 2 terms, pits and plasmodesmata expanding. Pits normally means a gap or weak spot. In the case of cell wall, it is not uniform in thickness throughout, because in certain areas, secondary wall is not laid down. Naturally, these areas are unthickened and are called pits. Normally, pits of adjacent cells will be placed opposite, this way, opposite. to each other form pit pairs. Pits with a uniform diameter is referred to be simple pit. Now comes bordered pit. Bordered pit cavity is flask shape. Example is tracheids. Now, plasmodesmata is the living component in the dead cell. It's an important item, dead cell wall. So once again, plasmodesmata is the living component in the dead cell wall. Dead cell wall. Many plasmodesmata or cytoplasmic strands are seen in pits by which cytoplasm of adjacent cells will be getting contacted. These cells are lined by cell membrane and contains a fine tubule, desmotubule. Again, you can observe this diagram or NCERT. Many scientists suggest origin of plasmodesmata may be linked with the endoplasmic reticulum. It is a part of simplast system between the two cells, inner side connection, simplast, apoplast, outer side. That is regarding the plasmodesmata and pits. Now comes the last topic, just an introduction, endomembrane system introduction. In a cell, each of the membranous organelles is distinct in terms of its structure and function. Many of these are considered together as an endomembrane system. Why? Because their functions are coordinated. And what are the main endomembrane system organelles we have to learn? That is the next episode. Endoplasmic reticulum, short form ECR, Golgi complex GB, then or Golgi body, lysosome, and vacuoles. So endomembrane system will be continuing next episode. After that, you can find 5 theory question, 5 NEET model question with the key. So we can just revise the topic, what all things we learned in this episode. So cell, the unit of life, episode number 3, where we'll be mainly dealing with the eukaryotic cell membrane, cell wall, endomembrane system introduction and question. Introduction, comparing with the prokaryotic cell, what are the features of eukaryotic cell, what all things or organism will be there with the eukaryotic nature from a connect with the NCERT page number 91. Then general characters of eukaryote theory question. After that, all eukaryotes are not identical. Plant cells and animal cells are different. Now, plant cell, what all things they possess, animal cell, what all things they possess, and to NCERT diagram, plant cell, animal cell. Then we entered into the famous structure cell membrane because it is seen in all living cell. And up to 1950s, then 1950s, we have no much idea about cell membrane. After that, we got a clear-cut idea because discovery of endoplasmic means electron microscope along with it. That is mid 1950s or 1950s. That is the year usually we say. Chemical studies proposed or performed and structure by analyzing human RBC, mainly lipids and proteins, and lipids, phospholipids, then arranged in bilayer, then polar head, hydrophilic, non-polar tail, hydrophobic. Then next is how it interacts with water and why that structure has been given. Then we came to a famous term, amphipathic nature. Amphi means both. That is, both hydrophilic and hydrophobic regions will be there. Connect with the polar, non-polar environment, play a crucial role in biological system, cell membrane. After that, some structures, photographs, then different models. Then thickness of the cell membrane, 75 Armstrong, based on J.D. Robertson. That is protein, lipid protein, how it becomes 75. Then different diagrams given the different models. Then again, a detailed study, some diagrams not in NCERT, old NCERT. One is David Robertson, unit membrane, 1959. Then trilaminar concept sandwich model, that is Davison and Danieli. Then famous fluid mosaic model, Singer and Carlson, that year is important, that is 1972. After that, different models. Then each case, proteins and lipids. Ratio of proteins and lipids varies considerably in different cell type. And there is a famous phrase, normally protein iceberg in a sea of lipids. So proteins will be less, lipid will be more. But in human RPC, erythrocyte approximately more, 52% protein and 40% lipid. Depending on ease of extraction, 2 types of proteins, peripheral and integral proteins. Then we came to that Singer-Nicolson's model, important for exam theory, all this, fluid mosaic model. Then quasi-fluid nature of lipids enables lateral movements of proteins within overall bilayer. The ability to move within the membrane is measured as its fluidity. Quasi means semi, NCERT page number 94. Then peripheral proteins also seen on periphery. That is, the nature peripheral proteins are seen on periphery. That is how the term came. On the surface of membrane, same time integral proteins are partially totally buried. Tunnel protein or integral protein, which run throughout the lipid bilayer, which is also called transmembrane proteins. Their removal is difficult, but it requires crude methods like treating with the detergents. And these two showing lateral movements and flip-flop movements. Proteins rarely flip-flop across a cell membrane. Lipids can flip-flop better than proteins. Then we entered into importance of fluid nature of membrane, explaining endocytosis. Pino and Fago. Then passive transport based on concentration gradient and osmosis, diffusion, it proceeds. Then facilitated transport due to some obstacles or obstruction, it needs a carrier. That is, polar molecule cannot pass through non-polar lipid layer, that is facilitated. Active transport, uphill transport, that requires energy, ATP utilized, and sodium-potassium pump in animal cell. There we observed a statement, one ATP molecule is used to move 3 sodium ions out of the cell, extracellular, and 2 potassium ions into the cell, intracellular, crucial for various cellular activities, nerve impulse, transmission, etc. Then came to cell wall. Cell wall is structure that's seen outside the membrane, non-living, rigid structure. It is seen in plant, fungus, bacteria, and algae. And there different chemicals, plant cell, which are the material for cell wall, algae, fungi, bacteria. Then in a plant cell from outside to inside between two cells, how it will be arranged. The junction of two cells, middle lamellae, then inside it primary wall, inside it secondary wall if present, then inside is the cell membrane closest to cytoplasm. Then next is primary wall. Young plant cell we can find single layer, elastic, thin, and gradually that is able to expand while growing, able to grow by addition of more wall materials within the existing one. Typical example, parenchyma cells and mesometic cells have only primary cell wall. Then secondary wall. As the cell matures, internal to primary wall, more layers of wall material will be added. Primary wall will be gradually diminished. Then typical example deposition like chemicals, lignin, superin, harder woody parts. Then middle lamellae, it is famous question. The cementing material between two plant cell made by calcium and magnesium pectate. When fruit ripens, it becomes soft. Then next is plasmodesmata. Again, an old NCERT diagram. Showing plasmodesmata, then functions of cell wall. After that, pits and plasmodesmata. Pits means gap or weak spot. Paired pits can be seen. Then pits pairs. Then pits with a uniform diameter is called simple pits. Then in bordered pit cavity is flask-shaped, example trachea. Then plasmodesmata is a living component in the dead wall. Many plasmodesmata or cytoplasmic strands are seen in pits by which cytoplasm of adjacent cells contacts. And these are lined by cell membrane and contains a fine tubule called desmotubule, diagram given here. Then it helps connect up with a symplastic system between two cells. Many scientists suggest origin of plasmodesmata may be linked with ER. Now, endomembrane system introduction, why they are called endomembrane system and which are the cell organelles present in them. That will be continuing next episode. After that, 5 theory question, 5 NEET model question with the key. If you like this episode, subscribe or like it, share it, comment on it. Meanwhile, short question I'll be daily putting. So for time being, we can wind up. Thank you. Take care. Bye.